Hi, hello. So we continue our survey on Oedogonium. As we have already seen, Oedogonium is a filamentous unbranched freshwater algae which belongs to the division Chlorophyta. We have seen its thallus structure. It is having three different kinds of cell: apical cell, basal cell, and in between intercalary cells. We have discussed the general thallus structure and asexual reproduction in Oedogonium in our previous lectures. Today we are going to see the sexual reproduction in Oedogonium. As I have already discussed, we can see that some intercalary cells bear cap-like structure. So that particular cells of the filament which bear these caps are called cap cells. Only these cap cells will be taking part in the reproduction, whether it is asexual reproduction or sexual reproduction. So what happens during sexual reproduction? During sexual reproduction, any of the filamental cells, any of the cell, let us take this cell, it is having a proper nucleus and cytoplasm. Any of such a cell which is having a cap will be doing segmentation of the cell. Okay, it will be having transverse divisions. See, like this: one division, two division, then multiple division. You can see that the nucleus also divides in that many number. Normally during sexual reproduction, 2 to 40 such segmentation will happen in one particular cell. Okay, 2 to 40 such segmentation will happen. And what is the time of the sexual reproduction? It is during the unfavorable situations. What are the unfavorable situations? Maybe deficiency of nitrogen in the water, lack of adequate light, etc. Or maybe even increase in the concentration of carbon dioxide in the aquatic body. During this time, any of the cap bearing cell will be doing transverse division, and it will cause 2 to 40 such segmentation. That means 2 to 40 antheridial cells. Each of these segments are called antheridial. Okay, so each of the antheridia will be producing 2 anthrozoids. Okay, it is initially one nuclei which got inserted into each antheridia. This antheridia during maturity, this particular antheridial nuclei will divide into 2, and each nuclei will carry some cytoplasm with itself, and it will form this pear-shaped anthrozoid. As soon as the maturity is reached, this particular antheridia at the junction of the cap will split open and will release these 2 anthrozoids which are covered in a vesicle. Okay, vesicle will dissolve soon after its contact with water. So you can see that around 2 to 40 such segments or antheridia got created from a single cell, and one particular thallus of Oedogonium will be having many of such cells. So from the cap bearing cell, it got segmented into around 2 to 40 numbers, and each of the antheridia will be releasing 2 anthrozoids. For example, if you are taking a number called 40, for example 40 such segmentations are there, we are taking the maximum number, and in each of these 40 antheridia there will be release of 2 of the anthrozoids. So 40 into 2, 80, maximum 80 such pear-shaped anthrozoids can be released from one particular cell of the Oedogonium. So just imagine how many number of anthrozoids will be created if there are multiple such cells taking part in the reproduction process. So this is one thing, and we have released the anthrozoids to the water. Now, these particular Oedogonium species can be homothallic or heterothallic, or monoecious or dioecious. In homothallic or monoecious condition, both antheridia and oogonia, or the female reproductive structure, will be formed in a single thalli. In the case of dioecious or heteroecious Oedogonial filament, there will be 2 separate thalli or 2 separate filaments for antheridia and oogonia. Okay. For example, in one particular kind of like Oedogonium called Oedogonium fragile, Okay, there will be monoecious situation, and in the case of Oedogonium aquaticum, you can see dioecious thalate condition. So that is one thing. Now anyway, we are taking the case of, we are just considering the Antheridia and Oogonia. We are not considering whether it is in single filament or like two different filaments. So antheridial development we have already seen. Now coming to Archegonia, Oogonium. Okay, in Oogonia, it's not Archegonium, which is the correct terminology is Oogonium. So during maturity, just like one of the antheridial filament, one of the cell of the antheridial filament got converted into antheridia through segmentation. During maturity, this particular cell or any of the cap bearing cell of the oogonial filament will be converting itself into a oogonia. You can see that no such segmentation is happening in the case of oogonial development. It is becoming bulged. It is just getting bulged. So this is the thing that is happening. Now this particular cap bearing oogonium will be transformed. Actually it was initially a normal cell of the antheridial filament. Now during maturity it got transformed itself into oogonial mother cell. Okay, you can write such a step initially, oogonial mother cell. Then from this oogonial mother cell it will undergo transverse division to form an upper oogonium and a lower suffoliatory or supporting cell. It's not like segmentation that is happening to create multiple antheridia, but it will be in this form. It was initially a single cell. Now the single cell got divided into two, just one division. Okay, one transverse division to create the upper oogonium and the lower suffoliatory cell or supporting cell. Okay, you can see this. Now what will happen? This particular oogonium at its maturity will be creating a slit for the entry of anthrozoids. Now we have already discussed the particular release of anthrozoids into the aquatic body. Now these pear-shaped anthrozoids, which is having flagella to swim, it will be swimming and coming to enter through the pore or slit which is present in the oogonium. It will enter and it will fertilize this particular egg that is present within the oogonium. So this is about the general reproductive feature of Oedogonium species which is happening. Now we have to see that there are two different types of species are present in Oedogonium based on the distribution of Antheridium and Oogonium. Okay, you can see that this particular, we have already discussed that there are around 400 species of Oedogonium present. Based on the distribution of Antheridia and Oogonia, we have classified them into two types, macrandrous and nanandrous type. Okay, actually the basic difference is in the formation of Antheridia. We will see what is the modus of operandi in this particular macrandrous species and nanandrous species. First, let us take the case of macrandrous species. What will happen? Initially, just consider the macrandrous species. For that, we can go to the previous slide. Okay, just consider this as the macrandrous species. Everything is happening normally, just like we have discussed the general sexual reproductory phase. This is the thing that is happening in the macrandrous type of reproduction. Okay, this particular thing that we have explained is the macrandrous species. Now we will see what is happening in nanandrous species. You just see this line. Okay, you just see this line. The top line, we have the row on the top. We have already explained regarding macrandrous species. Now just see the second row. This is the development of nanandrous species or nanandrous mode of reproduction. Just see, in nanandrous species, it is always dioecious. In macrandrous reproduction, we have discussed that it can be either monoecious or dioecious, but in nanandrous species of Oedogonium, always the Antheridia and Oogonia will be developed in different thalli. So let us see how that particular transformation takes place. In nanandrous species, during the reproductory phase, one particular cell of the Oedogonial filament will transform itself into androspore releasing body, or it can be called androsporangia. This particular cell of the Oedogonium thalli got transformed itself into androsporangia through transverse division. Okay, there will be multiple division and each will be forming androsporangia which will be bearing androspore. But unlike in the case of Macrantha species, you see the Macrantha species above, it was developing around two, it's not around, it was developing two androzoids within each antheridia. But here it will be developing androsporangia. Instead of antheridia, it has already developed androsporangia, and from that, instead of two antheridia, instead of two androzoids, as in the case of Macrantha species, it has developed one single androspore. Since the body is called androsporangia, it will be bearing androspore. This androspore will be released by a split opening up of this particular androsporangia, and this androspore will be swimming to reach the oogonial supporting cell in the other filament. I'll show another figure for this, which will give more clarity. Yeah, you see, this particular androspore will be moving to a different thalli where oogonium is there, and it will not be, it can either attach directly to the oogonium or it will be attaching to the supporting cell or suffragatory cell which is beneath the oogonium. Now what will happen? Why after attachment of this androspore, antheridial male filament or a small dwarf male filament will be developed by the germination of this androspore. For that, what will happen? This androspore after attachment will be dividing into two. That bottom cell will be forming a holdfast, and the top cell will be doing segmented division, and the top area will be bearing antheridia, multiple antheridia, because this particular androspore after germination, it is dividing once into two, and the bottom post area will be forming a basal cell or holdfast, and the top cell will be forming like a filament which is having 2 to 4 number of cells. So this small filament is known as nanandrium or dwarf male filament. This dwarf male filament will be dividing or segmenting its cells to form multiple antheridia, and each antheridia will be releasing only one particular androzoid. Okay, one particular androzoid. This androzoid will be moving towards the slit opening of the oogonia, and this will be fertilizing the egg within. Okay, now what will happen? It will be forming a zygote, and the particular zygote or oospore, which will be undergoing meiosis before its germination, and that particular zygote will be forming 4 haploid zoospores, because zygote was diploid. After germination, it will be releasing 4 haploid zoospores. These particular zoospores will be, each of the particular zoospores, it will be having a hyaline area, just like we have seen in asexual reproduction. At its tip, it will be having a clear or translucent hyaline area which is surrounded by basal granules from which flagella arises, and it will be attaching to a substratum. From this substratum, from after attachment, new filaments will be formed. Okay, the basal hyaline area will be forming the holdfast or for attachment. Then the top area will be forming the proper new thalli of Oedogonium. So this is all about the Macrantha and Nanandrous reproduction in Oedogonium. So this is the notes, or I have explained each and everything of these notes. You can pause the video and take down the notes. So this is the explanation. It is continuing, and here I have explained everything regarding to the Macrantha and Nanandrous type of reproduction. So this is all about today's session. So with that, we have completed the 3 parts of Oedogonium. First, we have introduced the structure of Oedogonium. Then we have, as a part of that video itself, we have discussed the general features and vegetative reproduction. Then in another lecture, we have explained the asexual reproduction in Oedogonium. Now we have discussed the general sexual reproduction and the Macrantha and Nanandrous type of reproduction. You can see that the drawings that I have attached are easy to draw drawings. So practice such drawing, and this question will be super easy for you to do in the examination. Thank you so much. In the upcoming videos, we will come with other genus life cycles. Thank you.